It's a pleasure to be with you. The title of this talk is Vaccine Safety Monitoring in the United States. Much of the content of this talk is adapted from the National Foundation of Infectious Disease Vaccine Safety Monitoring webinar presented by Dr. Daniel Salmon from October 2019. There are no disclosures for this presentation. Objectives include review key background information regarding vaccine adverse events, review the 4 basic components of the vaccine monitoring system in the United States, discuss the 4 phases of clinical trials and how they relate to vaccine safety issues, discuss passive, active, and clinical aspects of post-licensure vaccine safety surveillance, and review the main agencies associated with the federal vaccine system and their functions. This slide lists some key goals for vaccine monitoring programs, including ensuring that vaccine benefits outweigh risks for individuals and populations. Vaccine standards are high since vaccines are generally given to healthy individuals, adults and children, as a primary prevention measure. This is a different standard than intervention treatments like chemotherapy for cancer or antibiotics for individuals with disease. The benefits must clearly and substantially outweigh the risks in primary prevention programs like immunizations. Monitoring provides information that informs vaccine strategies to optimize the use of limited resources. Monitoring also encourages public confidence that ultimately results in improved vaccine coverage. This slide demonstrates a common evolution of an immunization program through 5 stages. In the pre-vaccine stage, disease is high since no vaccine is available. With vaccine introduction and immunization program strategies implemented, vaccine coverage increases and vaccine-associated disease dramatically decreases. With vaccine usage, adverse reactions, which are commonly relatively rare and mild versus disease risks, increase, but due to enhanced public concerns about adverse reactions as disease incidence declines, public confidence wanes and vaccine coverage declines. In some instances, adverse reactions become more common than the incidence of disease. A good historical example of this was seen with live attenuated oral polio vaccine. With a disease outbreak often exacerbated by less vaccine coverage, public concern and confidence commonly rebounds, resulting in increased vaccination rates. This cycle commonly repeats, as has been demonstrated in many countries with various vaccine-preventable diseases. The final goal is disease eradication, like that with smallpox, when no further vaccine is required and adverse reactions consequently eliminated. Adverse events following immunizations will occur. Some of these adverse events may be coincidental occurrences, and others, though rare and commonly mild, the result of immunizations, a causal adverse event. For example, as diagrammed on this slide, many individuals with underlying risk factors for diseases like heart disease and skin cancer, etc., will be immunized with various vaccines in populations like the United States. The daily incidence of myocardial infarction diagnoses in the U.S. is 3,000 per day, and for skin cancer, 1,357 per day. Relatively common clinical diagnoses in this country. As you might expect, whether given vaccines or not, these individuals may develop and be diagnosed with skin cancer or having a myocardial infarction, MI, since the causes of these diseases are complex and multifactorial. A major vaccine safety monitoring question is whether the vaccine was a coincidental or causal factor for adverse events like these. Many anti-immunization groups use coincidental events as evidence that vaccinations are dangerous, arguing incorrectly that the order of events proves causality. The differentiation between coincidence and causality requires robust epidemiologic follow-up and analysis. This is the mission of vaccine monitoring systems. This slide lists some contemporary issues that impact vaccine programs and the public's concerns about vaccine safety. The emerging medical model of shared decision-making. This means that clinicians must understand vaccine details and be able to adequately and effectively discuss vaccinations with their patients and families in the face of even more limited patient time in the clinical setting. Media focus on sensationalism and the exploding misinformation provided via social media enhances public's distrust of vaccinations and corporations associated with the vaccine system. As a result of sensational reporting, social media misinformation, and the resultant public mistrust of vaccine experts, parents claiming school vaccine exemptions for their children is rising in many states. For example, kindergartners in Texas must have 10 immunizations to be enrolled in school. Texas first started reporting vaccine exemption data in 2006. The exemption rate for kindergartners in Texas has risen from 0.3% for the 2005-06 school year to 2.15% for the 2018-19 school year. Also, increased rates of parents delaying or refusing vaccinations has been noted from several studies from 2012 to 2017, showing that 23% of parents delayed vaccinations for their children who are less than 5 years old, and 16.7% of parents refused one or more ACIP recommended vaccines for their children. Parental vaccine behaviors have been negatively influenced by misinformation. 77% of parents report vaccine concerns, including 34 to 38% concerned about too many shots for their children, 16 to 32% concerned about safety issues, including vaccines causing fevers, autism, chronic diseases, etc. 11% stating that it's unlikely that their child will get vaccine-covered diseases, and 8% believing that vaccine-associated diseases are not serious. These concerns can be addressed by effective vaccine safety educational strategies for parents. This is a classic bell-shaped curve that represents the distribution of biologic response to immunizations in the population. As illustrated, there are high and low responders. The high responders are generally more reactogenic and therefore tend to exhibit more adverse reactions than the low responder group. These high responders are generally the focus of vaccine safety monitoring programs. This slide lists the 4 basic components of the vaccine monitoring system in the United States. 1. Clinical trials. 2. The licensure process. 3. Post-licensure surveillance. And 4. Causality assessments. The four components do not operate in isolation. There is significant overlap of activities, as will be demonstrated in the following slides. The post-licensure surveillance process includes passive, active, and clinical assessment surveillance. Let's consider the four phases of human clinical trials and licensure of drugs and vaccines for a few minutes. The human studies are commonly preceded by extensive non-human and laboratory studies, resulting in hypotheses that set the stage for human clinical trials. Phase 1 clinical trials focus on the study agent in a relatively small number of healthy subjects, approximately 20 to 100 in number. The goals of Phase 1 trials include looking for safety issues, including side effects, and evaluating how the agent should be taken. In vaccines, this includes the gathering of dose-related immune response data. Phase 2 trials also evaluate the study agent for safety, but in a larger study group, usually a few hundred up to 1,000 in number. For a medication study, patients are those with the condition for which the medicine, device, product, or treatment has been developed. For vaccines, the study group is generally healthy individuals. Researchers in Phase 2 trials assess the short-term safety and effectiveness of the drug or vaccine, explore drug or vaccine interactions, and find the dose at which the agent works best with the least side effects, and conduct a small-scale placebo comparison. Phase 3 trials look at a larger group of individuals, often 1,000 to 20,000 individuals that better represent the health of the population. Phase 3 trials aim at confirming safety of the agent, evaluate efficacy in this larger population group, compare the new agent to other compounds, for example, versus a placebo or other therapies, define risk benefits, and gather information for a licensure application and package insert. Following the completion of Phase 3, a drug or vaccine is approved or not approved by the U.S. Food and Drug Administration, FDA, for use by specific patients. Phase 4 may only begin if the drug is approved. 12% of medicines that are evaluated in clinical trials will be licensed. Finally, after the drug is approved, researchers look to study the effectiveness of the drug in a wide variety of patients, as well as monitor the safety in a large group and enable the development of new uses for the compound. These studies often involve thousands of individuals from the population using the licensed drug or vaccine. Phase 4 studies often inform future research and development. Phase 1 to 3 trials are often not of sufficient numbers to appropriately evaluate intermediate or rare adverse events. Due to the large number of study subjects, Phase 4 studies can expand the safety profile of the drug or vaccine by evaluating that larger study group for intermediate or rare adverse events, along with safety in subpopulations. This phase of studies also address any emerging safety issues that arise with drug or vaccine usage. As mentioned previously, Phase 4 studies relate to post-licensure safety monitoring, where Phase 1 to 3 studies address the pre-licensure and licensure safety monitoring process. This slide lists the advantages and limitations of Phase 1 to 3 clinical trials. Advantages of Phase 1 to 3 clinical trials include the use of double-blind randomized studies to reduce confounding and bias, These trials use strict inclusion and exclusion criteria for participants to reduce participant risk and reduce confounding. Double-blind randomized trials also use incremental studies to minimize risk. Phase 1 to 3 clinical trials are associated with several limitations related to safety of drugs and vaccines. These trials can't evaluate adverse events in persons excluded from a study, like those excluded due to their use of other medications, underlying health conditions, etc. They also can't adequately evaluate delayed adverse events outside the study period, rare adverse events due to small sample size, and adverse events in subpopulations. This underlines the importance of post-phase 1 to 3 clinical trial vaccine safety monitoring to address these limitations. The epidemiologic calculations for this table are complicated and beyond the scope of this talk. This table demonstrates the sample size needed to detect various rates of rare adverse events. The main point is that very large sample sizes would be required of phase 1 to 3 clinical trials to detect rare adverse events. This emphasizes the importance of robust phase 4 clinical studies and other post-licensure vaccine safety monitoring systems to detect these rare safety issues in general population vaccination programs. We've already discussed the role of phase 4 clinical studies in post-licensure vaccine safety surveillance. For the next few minutes, let's consider a few more details regarding the post-licensure surveillance process. Post-licensure surveillance includes 3 surveillance categories: passive, active, and clinical assessment. This slide lists the various U.S. programs involved in vaccine safety post-licensure surveillance. Passive surveillance is addressed by the Vaccine Adverse Event Reporting System, VAERS. Active surveillance relates to the post-licensure Rapid Immunization Safety Monitoring Program, PRISM, a Food and Drug Administration FDA program. The Vaccine Safety Data Link, VSD, a Centers for Disease Control and Prevention, CDC program. The Centers of Medicare and Medicaid Services, CMS, the Indian Health Service, IHS, the Department of Defense, DoD, and the Department of Veterans Affairs, VA. There is a Federal Immunization Task Force, ISTF, that includes representatives from all agencies within the U.S. Department of Health and Human Services, HHS, with assets in vaccine safety, including the FDA, CDC, CMS, and IHS, along with the Department of Defense and Department of Veterans Affairs. Let's discuss passive surveillance and the Vaccine Adverse Event Reporting System, VAERS, for a few minutes. The purpose of passive surveillance like VAERS is to detect signals of unanticipated events that deserve further follow-up. Passive surveillance needs to be highly sensitive. High sensitivity is commonly associated with low specificity. Highly sensitive means that all real events will be picked up. Low specificity means that there will be many false positive events identified. There are many limitations for passive surveillance. Passive surveillance generally can't determine cause from coincident events. For epidemiology and biostatistics, passive surveillance data commonly lacks good denominators and comparison data like an unvaccinated comparison group. Passive surveillance data often suffers from bias, including underreporting, incomplete reporting, overreporting, etc. VAERS is a passive surveillance program that accepts reports from anyone. It's co-administered by the Centers for Disease Control and Prevention and the Food and Drug Administration. Systems like VAERS have the potential for high sensitivity by not missing real events. Yet there is also a great risk for data bias that can result in limitations of under or incomplete reporting, which is common with VAERS. Passive surveillance systems like VAERS can't determine causality due to incomplete data and the lack of appropriate comparison groups like unvaccinated individuals. VAERS was designed to detect signals and help develop hypotheses for further studies to determine causality. This diagram illustrates the limitations of VAERS data, which only provides the number of people reporting an event after receiving a vaccine. It provides no data on the adverse events in those not vaccinated and those who were vaccinated but did not have the adverse event. VAERS data generally can only be used to create hypotheses for further studies. This quote from the National Vaccine Information Center demonstrates the limitation of adverse event underreporting through passive surveillance systems like VAERS. There have been estimates that fewer than 10%, even as low as 1 to 4%, of adverse events which occur after prescription drug or vaccine use are ever reported to government adverse event reporting systems. If only 1 to 4% of all adverse events associated with human papilloma virus HPV vaccination are being reported to VAERS, there could have been up to 38,000 health problems, adverse events after HPV vaccination in 2006, which were never reported. Let's now consider active surveillance systems in the U.S. The Vaccine Safety Data Link VSD program is an active safety monitoring tool. It was developed by CDC approximately 30 years ago. It covers 9 million people linked through managed care organizations, MCOs. Data includes vaccinations, hospital and outpatient information, and laboratory data. VSD utilizes rapid cycle analysis, RCA, which was developed by VSD to monitor pre-specified vaccine-related safety outcomes in near real time. Rapid cycle analysis commonly utilizes either self-controlled or current historical study designs with appropriate control groups, data quality, and adjustment for confounding factors. Every week, RCA compares rates of pre-specified outcomes in pre-specified time windows. 90% of initial adverse event signals turn out to be spurious by rapid cycle analysis. So initial adverse event signals from initial surveillance processes are not very specific. These initial signals are false positive 90% of the time. This is an example of pre-specified rapid cycle analysis outcomes from the 2009-10 influenza season, the year of the H1N1 pandemic. Let's now discuss briefly the post-licensure rapid immunization safety monitoring PRISM system. PRISM was developed by the National Vaccine Program Office for the 2009-10 H1N1 season and has been ongoing since then, providing data to the Food and Drug Administration. The PRISM system captures information from 8 state immunization registries and 4 large health plans, placing 107 million people, or approximately 1/3 of the population of the United States, under active surveillance. PRISM uses rapid cycle analysis and ad hoc studies to assess vaccine safety data. This table reviews the federal departments and agencies that provide services for focus groups and perform active vaccine safety surveillance, including the Centers for Medicare and Medicaid Services, CMS, the Department of Defense, DoD, the Department of Veterans Affairs, VA, and Indian Health Service, IHS. CMS covers a variety of age groups, though predominantly individuals 65 years old and older, who receive approximately 14 million vaccinations annually. The DoD provides services to approximately 2.6 million individuals who are healthy adults. The VA covers about 5 million individuals who are mostly elderly and eligible federal employees who are not covered by the Department of Defense. Non-veterans health services are administered under the Office of the Chief Human Capital Officer, OCHCO. The Indian Health Service, IHS, covers approximately 350,000 individuals of all ages from 21 service sites. These systems can do chart reviews for data. Regarding vaccines, these systems predominantly focus on influenza vaccine administration and safety issues. Another program that provides post-licensure active surveillance is the Clinical Immunization Safety Assessment Network, CISA. Adverse vaccine events are rare occurrences for practicing clinicians. CISA attempts to provide a clinical support network to help practicing clinicians appropriately address potential vaccine-associated adverse events. CISA is a small network coordinated by the Centers for Disease Control that includes academic centers of excellence, vaccine safety clinicians, clinical subspecialty, and research laboratory resources. The goals of CISA include increase the clinical understanding of vaccine safety, associated nuances, utilizing scientific knowledge of immunology, pathophysiology, genetics, and predisposing risk factors, etc. Maximize the utility of VAERS as a disease registry, disseminate protocols and findings, and create a new clinical subspecialty of immunization safety. CISA provides real-time consultations and follow-up for clinicians utilizing standardized assessment and management protocols for persons with potential vaccine-associated adverse events. The fourth component of the U.S. vaccine monitoring system is causality assessment. The Institute of Medicine IOM Immunization Safety Review Committee is an entity that provides causality assessments. It was organized in response to an increased number of vaccine safety concerns and a growing level of public polarized vaccine beliefs. The Immunization Safety Review Committee provides a mechanism for timely, objective, and expert review of the strength of competing vaccine safety hypotheses. This committee evaluates vaccine adverse event evidence for causality and guides public health responses through research, surveillance, communications, and policy reviews. This slide lists some of the main agencies associated with the federal vaccine system and their functions. The National Institutes of Health, NIH, provide basic vaccine research, vaccine development, and oversee many clinical trials. The Agency for Healthcare Research and Quality, AHRQ, provides healthcare system delivery research. The Food and Drug Administration, FDA, oversees licensure, various inspection services, and post-licensure surveillance. The Centers for Disease Control and Prevention, CDC, provides post-licensure surveillance, clinical assessments, and communications. The Health Resources and Services Administration, HRSA, oversees vaccine compensation programs. In the federal system, there are several vaccine delivery programs and systems, including the Department of Defense, DoD, Centers for Medicaid and Medicare Services, CMS, Indian Health Service, IHS, Veterans Administration, VA, and the Office of the Assistant Secretary for Preparedness and Response, ASPR. The National Vaccine Program, NVP, provides coordination of the whole federal vaccine system. In summary, the United States has a robust vaccine safety system, including clinical trials, licensure, post-licensure surveillance, and causality assessments. The vaccine safety system involves several federal departments and agencies and non-federal partners. It's essential to investigate and address vaccine safety issues in a timely way.